Hello, everyone. A very warm welcome to my developmental biology class. Today we will discuss about fertilization in sea urchin. This is a very important topic from the Dev Bio unit. Okay, so let's get started. Here we will talk about the meeting of the sperm with the egg, and then we'll briefly talk about the mechanism of a fast block to polyspermy and slow block to polyspermy. Okay. Well, so you can identify this picture. This is a sea urchin. Sea urchins, as you all know, they belong to Echinodermata, the phylum Echinodermata mainly contains members which are marine in nature. Okay. So this creature is known to undergo external fertilization. External fertilization means what? You must be knowing by now what is external fertilization, right? External fertilization means the egg as well as the sperm are laid outside the body and they meet outside the body in the environment, right? So here sperm and egg meet in water, that means in the sea, right? Okay, so let's start the discussion. All introduction and all those parts about the organism and all, I'm not going into those details. You all know that. We'll quickly come into the topic. See, in sea urchin, you will see this meeting of the sperm with the egg is extremely specific. It occurs in a species specific manner. Okay. Fertilization of egg is species specific. Now, why do I say it's species specific? You see, in the marine environment, it's not only the sea urchin that is living in the ocean. There are plenty other organisms that also have external fertilization, right? So there are different types of eggs in the water and different types of corresponding sperms in the water in the open environment. So how do the sperm identify its corresponding egg? The egg that belongs to its own particular species. How will the sea urchin identify the egg from another sea urchin? This is called species specific recognition. Okay. So what happens? See, the egg vitelline membrane has receptors. Okay. It has got receptors on on the membrane. These receptors are called EBR1. The egg vitelline membrane has got receptors on its surface. What is the name of the receptor? EBR1. These receptors are extremely specific to this organism. This EBR1 receptor is meant for a specific ligand, which is there in the sperm. What is the ligand for this? Sperm has the ligand protein. What is the name of that? Bindin. Right? So this is one part. Next you see, next we have another very important protein from the egg. This protein is known as resac. This resac protein is a 14 amino acid protein, right? This is a chemoattractant which is released by the egg and it provides direction to the sperm. So the sperm knows which way it should swim, in which way it should travel in order to meet the egg. So this resac protein gives it a proper direction in which it should swim in order to unite with the egg. Clear? So this resac is a chemoattractant that provides direction to the sperm. Clear? So far, it is nothing difficult. Fine. Now see, this is a protein. As I said, resac is a protein. This will also have a receptor. Here we had talked about, see, one second. Look, here we have talked about the ligand being present in the sperm and the receptor being present in the egg. Now the case is just opposite. The ligand is in the egg, which is resac. The receptor for this is in the sperm. The sperm has the receptor. What is the name of the receptor? The name is receptor guanyl cyclase. RGC. What is the full name? Receptor guanyl cyclase. Fine. Now what happens here? Here the GTP gets converted to cyclic GMP from the very word cyclase. You can understand what is happening. So the GTP here gets converted to cyclic GMP, which activates calcium channel. Fine, it will activate the calcium channel. You will understand this later as we discuss it in more detail. You will understand this very clearly. Now, what will happen? An acrosomal process will be formed and bindin gets expressed all along it, means a structure like this. You know, the sperm is somewhat like this, right? You have seen here, see? Here you have seen, no? This is how the sperm looks like. Now, when acrosomal process will be formed, a structure which is somewhat like this will develop and bindin will be expressed all along this process, right? And along with this, what will happen? Other acrosomal enzymes, which are proteolytic in nature, they will start to digest the jelly part of the egg surface so that it can penetrate and, it can uh, facilitate the process of fertilization. Clear? So what did we learn here? The two most important things. One receptor from egg and its corresponding ligand in the sperm, that is EBR1 and bindin. Next is one protein from egg, that is RESAT, which is a chemoattractant and gives direction to the sperm, and its corresponding receptor on the sperm, which is known as RCG, which is the name of it is receptor guanyl cyclase. Clear? So this is what is actually happening. Now, once it comes near the egg, it will digest the jelly part of the egg surface and all those stuff it will do, and it will fertilize the egg. Now, see, one sperm will fertilize the egg. Polyspermy is not at all going to happen. If that happens, what will happen? It is a lethal condition. It is a lethal condition, and it will give rise to polyploidy and all those stuff. So it is not at all going to happen. Okay. So there is a block mechanism that operates, which will prevent a second sperm to come if one sperm is successfully fertilizing the second one will never get a chance. So before we get into that, you first take a look at this picture. What happens? See, this is the sperm. It shows the fusion of the sperm plasma membrane with the acrosomal membrane and release of acrosomal content. The acrosomal content gets released. Fine. See, look here. What happens? This is the nucleus part. This is your acrosomal membrane, and this is the bindin, which we were talking about, whose receptor is the TBR1. Fine. Next, what you see here, see, in this one A, B, and C, what you get to see here, you get to see the fusion of sperm plasma membrane with the acrosomal membrane and the release of the acrosomal content. Acrosomal content means what? As I just now told you, acrosomal enzymes, which are proteolytic in nature, and these will digest the jelly part of the egg surface. Clear? Now see. Now, the actin molecule will assemble to produce actin filament. Okay, the actin part, this actin molecule will assemble to produce actin filament. And this, as I told you, a process will form, acrosomal process will form around which or along which, see this part, along which bindin gets expressed. All along its surface, bindin will get expressed. Clear? So what happens here? What is the end picture, end story? Bindin gets expressed all along the acrosomal process. Clear? Nothing difficult so far? It's easy? Okay. So now we will see what's happening after the sperm successfully meets the egg. As one sperm is meeting the egg, it is not going to allow the second one to come. Clear? So this polyspermy is blocked by two mechanisms. Polyspermy is blocked by two mechanisms. One is slow block to polyspermy, the other is fast block to polyspermy. Well, we should write fast block to polyspermy first and then we'll write slow block to polyspermy. Fine. First is the fast block to polyspermy, then you come to slow block to polyspermy. The fast block to polyspermy is also known as electrical block. Okay. Electrical block. Voltage is involved here. Clear? Electrical block. The slow one is known as calcium mediated block. Calcium mediated block. Clear? So fast block to polyspermy is also known as electrical block, and the second one, slow block to polyspermy, is also known as calcium mediated block. Clear? Now we will see one by one in detail. Fine. Before you learn about the slow and fast and slow block, just take a look at this ones. See, this is a nice picture and it is labeled very nicely. I mean, sequentially it is labeled. First, see what happens. This is the first one. You just see the egg comes very near to it. The content is yet not released. In the second step, what happens as it comes here? The content, the hydrolytic enzymes and all, it comes out and the actin filament forms here. It forms, the actin molecules assemble and it gives rise to the acrosomal process, and this bindin molecules are all expressed around it. Along with it, all these happens. Huh? Then you see, you move here and you see this. These changes that you get to see here will be discussed while we discuss the fast and slow blocks. Fine. So let's start with fast block to polyspermy. Clear? Remember, this starts fast block to polyspermy. It starts within 2 to 3 seconds of fertilization. That is why we call it fast. It's very fast. 2 to 3 seconds of fertilization. There is no delay. Immediately it kicks off. The process gets kicked off. It starts. What happens here? See, that is important. The membrane potential of the egg is negative. The membrane potential of the egg, remember, of the egg is negative. Clear? Now, this negative membrane potential is very important for the sperm to come and fuse with the egg. Clear? Now, since the egg has already got fertilized, so what will it do? It will change. There will be some change in the membrane potential, and this negative membrane potential changes to positive. It is initially say minus 70 millivolts, something around minus 70 millivolts. This changes to hmm, change in membrane potential changes to 20 millivolt. This is not at all uh, required for sperm binding. Hmm? Sperm doesn't need a positive membrane, it needs a negative membrane. So, this changes this minus 70 millivolt changes to plus 20 millivolt. How is it happening? The it changes the membrane potential of the egg as influx of sodium ion from the sea occurs. See, I told you it is external. Hmm? From the sea, it is happening in the sea, and you know, sea water is salty, content of sodium is high. So there is an influx of this happening because of influx of sodium ion. Clear? This is happening because of influx of sodium ion. Now, as the membrane becomes positively charged, sperm can no longer bind to it. I told you negative membrane potential is necessary for fertilization. This is short-lived, and very soon the membrane potential reverts back to negative. Now, this changed membrane potential is short-lived. This is short-lived, maybe for 30 to 40 seconds. This is short-lived. Now, what will happen after this? It will again revert back to negative. Now, as it reverts back to negative, you can say that, ma'am, there are high chances of a new fresh sperm to come and bind to it. Yes, there is a chance. That is why at this point, slow block comes into play, right? Fast block is just very, very temporary. Now this slow block is coming into play. What is happening in slow block? Slow block is also known as calcium mediated block. Slow block is also known as calcium mediated block. It occurs after fast block. Here you see what happens. You have heard of the enzyme PLC? PLC, phospholipase C. Hmm? Phospholipase C cleaves IP2, I mean the phospholipids, cleaves IP2 into, you know this, right? You know this, IP3 and DAG. So something which you already know. Clear? The released calcium. Hmm. Now what will happen? See, just right here. See, this is what is happening, right? Right? This IP3 will act on the calcium channels on endoplasmic reticulum of the egg. Hmm? It will cause release of stored calcium from the ER and increases agree. IP2 will be cleaved into IP3 and DAG with the help of which enzyme? PLC, phospholipase C. This IP3 will act on the calcium sensitive, calcium channels on the endoplasmic reticulum, and it will release stored calcium, and it will thus increase the calcium content in the egg. Now what will happen? The released calcium will activate. This released calcium, it will activate the cortical granules. This calcium will activate the cortical granules. The enzymes of the cortical granule will they will start to get released as the granules fuse with the egg membrane. Now see this cortical granules here. You can see the cortical granule. See these structures. These are the cortical granules. You can see here, I think it's a small little picture, but it has got a lot to say. See, these cortical granules start to fuse with the egg membrane. Clear? They start to get fused with the egg membrane. You can see this here. See, these cortical granules are here. Now you come here, this part. See, they are getting fused with the egg membrane. After getting released, it thickens the space between the vitelline membrane and cell membrane. Fine. And it uses a lot of oxygen to convert the outer membrane to fertilization envelope. Clear? And along with this, a hyaline layer is also formed, and the entire egg surface property changes. Remember this, the entire egg surface property is changing. So this influx of calcium helps in sperm, sorry. So this entire change in the property of the egg surface causes the causes the slow block to polyspermy. Had there been no release of calcium, this would have never happened. So the slow block is also known to be associated, or it's also called cortical granule mediated this block to polyspermy. Clear? Now, initially we had talked about this receptor of RESAC. You remember we said that the receptor is RGC, receptor guanyl cyclase, which is like GTP gets converted to cyclic GMP, which activates calcium channel along all those things. Now see what happens there. Influx of calcium there helps in sperm activation, and it helps the sperm to swim towards the egg. And once this happens, the sperm can move easily towards the egg. So calcium, see, plays a very important role in both. They play a very, very, very important role in both the egg as well as the sperm. Okay, so I hope this lecture is clear to you. It is nothing difficult actually. All that you need to remember is the name of the enzymes or proteins, its respective receptors, and slow and fast block to polyspermy. So before we wind up, in a nutshell, what happens here, you see sperm binding and or fusion with egg membrane. This happens. There is sodium influx. Hmm? This means what? Membrane potential change here. That means from minus 70 millivolts to plus 20 millivolts. This is what is happening, you know. This is called fast block to polyspermy. The other was which one? Phospholipase C comes into play here. It will it will cleave IP2 into IP3 and DAG, that is diacylglycerol production will happen. Fine. This will cause calcium release. Here, this will cause calcium release. This is very important. Very, very, very important. You just can't afford to forget it. And this will cause the exocytosis of cortical granules and it will form the hyaline layer. And this is causing slow block to polyspermy. Clear? Along with this, what is happening? NAD kinase is getting activated. It gets converted to NAD. It results into conversion of NAD to NADP plus. All these are happening. Other than this, what is this diacylglycerol doing? It is activating protein kinase A. Here, sodium hydrogen, this exchange is happening and it is causing a change in the intracellular pH. These together, the released calcium and this changed intracellular pH, causes stimulation of protein synthesis, DNA replication, and cytoplasmic movements of morphogenetic material. So this flow chart gives you an entire picture of egg activation in sea urchin. Okay, till then, winding up. Thank you so much for attending the lecture. If you have any confusion whatsoever, please feel free to get back to me in the comment section. I'll try my best to clear your doubts. Till then, happy learning and goodbye.